Okay, thank you. Okay, I guess we'll get started. Welcome back. Hope you had a good break. Hopefully you remember that last November we talked about reproductive function in males and females separately. And the basic concept, the big picture, was in males, the function was to make sperm and get them into the female. And the key steroid was testosterone. In females, it was more complicated because they had two systems. One was basically analogous to that in males. That was to make a mature ovum and ensure it's fertilized. And for that, the key steroid is estradiol. But they also had another function, which is what we'll be considering today. And that is that they had to maintain the fetus during pregnancy. And in that case, the key steroid was progesterone. So today we're going to put the two systems together and make a baby. This is obviously a very complicated process, so we're just really going to touch the surface of it and sort of highlight a lot of the key points. And basically, you can think of pregnancy and lactation as a series of steps. You start out with initiation of pregnancy, and then you go from, once it started, you have maintenance. Finally, you have delivery of the newborn infant, and then you have lactation. So today, the first lecture, we're going to consider initiation, and this can be divided into several sequential steps. The first is you obviously have to get the male and female together. That's a very complicated process known as courting. And ultimately, if you're going to have a baby, it culminates in sexual activity. You then have sperm delivery up to the oviduct to the ovum, and fertilization and implantation. So if we start with the first step, which is getting the male and female together, that involves the development of libido. In this case, the important hormones are androgens. These come from the male's intestines and the adrenals in females. Now, in most animals, maintenance, once you have libido developed, it's absolutely dependent on gonadal or steroids in adults, but in humans, that doesn't seem to be the case. If, for example, a male is castrated for some reason, some clinical reason, his libido will increase dramatically, but it will not necessarily go away completely. If we move on to the sexual act, despite the anatomical and behavioral differences between males and females, the underlying physiological events are fairly similar in the two. And it can be divided into 4 stages: an excitement or arousal, a plateau, orgasm, and resolution. Now, if we start with excitement, arousal, this can be initiated by a whole different types, a number of stimuli. They can be visual, auditory, tactile, or even psychogenic. But regardless of what initiates the process, the primary effect is to increase parasympathetic outflow to the genitals. This causes vasodilation, which starts the whole process. Note that the sympathetic nervous system is going to cause vasoconstriction. And that's one reason why stressful environments are not conducive to normal sexual function. Now, the increase in blood flow to the genital region has several effects. In terms of erectile tissue, the penis and clitoris, you get erection. In the female, you also get vasocongestion of the labia and vagina, and movement of fluid into the vagina. And the latter is important because that's going to involve the lubrication for intercourse. If we look at what's happening in erectile tissue, basically you have vasodilation, the blood flow increases into the sinusoid portion of the erectile tissue, and that increases the blood volume of that. Because of the structure of these sinusoids, this increase in blood volume constricts the venous outflow. So you get an increase in blood flow, but you get a decrease in outflow. So the net effect is you get a large increase in blood volume, and that's what causes the erection. Now, we now know a lot about the biochemical nature of this as well. As we said, this is caused by activity in parasympathetic nerves. but does not use a classical parasympathetic neurotransmitter acetylcholine. Instead, it uses nitric oxide. So you have action potentials coming down to the terminals. That causes an increase in calcium. Calcium activates NOS, and so you get an increase in nitric oxide synthesis, which diffuses rapidly into this vascular smooth muscle, increases cyclic GMP, and that's what causes the relaxation. Now, to terminate the whole process, there's an enzyme in this tissue. It's a specific phosphatidyliesterase. So that's going to break down cyclic GMP to GMP and reverse the relaxation effects. Now, this particular enzyme has a significant pharmacological significance because most of the drugs that are now available for erectile dysfunction, their mechanism of action is to inhibit this enzyme. So the net effect is, if you're taking one of these, your cyclic GMP levels go up higher, and they're maintained for a much longer period of time because they're not broken down as rapidly. So that's what's going on during the excitement phase. During the plateau phase, this is basically a continuation of the excitement stage. So you have continued parasympathetic outflow. A lot of this is due to reflex stimulation from a tactile stimulation in the genital region. In the male, that causes further vasocongestion of the penis, increasing its diameter somewhat, and the testes. In the female, you get vasocongestion of the outer third of the vagina. That's going to decrease the diameter, so the combination of those two increases the friction during intercourse. And the uterus moves up. This creates a pocket for the ejaculate can move into. And of course, you get a lot of increased systemic effects. Now, in contrast to the first two phases, orgasm is caused by a discharge of sympathetic nerves rather than parasympathetic nerves. This causes contraction of pelvic skeletal muscles and smooth muscles. In males, the smooth muscle contractions cause emission, which is movement of sperm and seminal fluid into the urethra. And in females, it causes contractions in the vagina and sometimes the uterus as well. The emission is almost always followed by ejaculation, and it's actually the volume of seminal fluid in the urethra triggers a local reflex that stimulates skeletal smooth muscle, and that causes ejaculation. And of course, you're getting maximum systemic and psychic effects during this period. Finally, you have resolution, and resolution is due to continued sympathetic outflow. And remember, what sympathetic nerves are going to do is they're going to cause vasoconstriction in the genital region. So the primary effect of that is to cause decreased vasocongestion and detumescence of the rectal tissue. And that's followed by a refractory period in males, but not in females. Okay, any questions? Okay, so we have the sperm delivered to the upper portion of the vagina, but they now have a ways to go before they can fertilize the ovum. The ovum's been recently ovulated, and it's up in the very top of the oviduct, the area called the ampulla. So the sperm have to get through the cervix, up to the top of the uterus, and then up the oviducts to the ovum. Now, one thing we know for sure is that they don't swim all the way because it's just too long for them to do that in a reasonable amount of time. But however, the first step, it does require them to swim. So they actually swim through the cervix to get into the lumen of the uterus. But after that, they rely on the female reproductive tract as sort of a transportation system. And this involves smooth muscle contractions in the uterine myometrium and smooth muscle contractions in the oviductal smooth muscle. So once they get into the lumen of the uterus, the contractions of the myometrium create sort of a churning action, so they're rapidly distributed throughout the lumen. And then there's what are called antiperistaltic smooth muscle contractions in the oviduct because they go up rather than down, and these aid their movement up to where the ovum's waiting. Now, there's an important aspect of this process, and it's dependent on the hormonal environment. If you remember some of our material we presented on the actions of estradiol and progesterone on the female reproductive tract, estradiol stimulates oviductal and uterine contractions and decreases viscosity of cervical mucus. So that's going to promote transport of the sperm because you want them to be able to get through the mucus fairly easily. So you want low viscosity, and then you have high smooth muscle activity. In contrast, progesterone inhibits all three. So the ideal time during the menstrual cycle for transfer of sperm is either late follicular phase or shortly after ovulation before progesterone levels go up when estrogen has been high for a while. Now, while the sperm are undergoing this transport, there's two important processes that occur. First of all, they become motile, so they can swim. Now, that's obviously essential because they have to swim through the cervix. And then they undergo a process known as capacitation. That's a sort of a fancy word that just means they gain the capacity to fertilize the ovum. So newly ejaculated sperm, if you stick them in with an ovum, it can't fertilize that. But during this time period, they undergo capacitation. Now, motility is gained simply by the process of ejaculation because you get dilution of the seminal fluid. So you get that in itself will increase motility, and you also get bicarbonate released in the seminal fluid, and the combination activates the sperm tail. Capacitation appears to involve a number of changes in the membrane, and it's time dependent, but it's not tissue dependent. So it doesn't have to occur in the oviduct or in the uterus. It can occur in vitro. Now, in terms of physiological importance of the seminal fluid, it's obviously not absolutely critical because you can take sperm from that are stored in males or in the testes for that matter and use them for in vitro fertilization. So you don't need seminal fluid at all. It is the primary volume of the ejaculate and appears to have two important roles that facilitate the process of fertilization. First, it has a lot of fructose, and this is going to provide energy for the sperm for the periods where the motility is critical. And they also have a number of buffers. And this also is important because the environment of the vagina is very acidic, and sperm don't do well in an acidic environment. So the buffers in the seminal fluid increase the pH and help the sperm survive until they can get into the uterus. Okay, so we've gotten the sperm up to the ovum, but it still has three more barriers to get through. The first of these is a cumulus oophoris, which are just some granulosa cells that have surrounded the ovum during its life in the follicle, and they're released with the ovum at fertilization. Now they're held together by a sticky protein, which is a protein conjugated to hyaluronic acid. The zona pellucida is a thick glycoprotein shell, and it's very important for survival of the ovum before fertilization because it protects the ovum as it's released during ovulation and picked up by the fimbriated end of the ovum. If you think about if there's only a plasma membrane surrounding the ovum, it would rupture very quickly during the process. It also protects the embryo as it travels down the oviduct into the uterus if fertilization occurs. So that's the second barrier. And finally, there's the ovum plasma membrane it has to get through. Now, the first barrier, getting through it, is accomplished by active swimming. So this is another step in the process where motility is critical. And the sperm have enzyme hyaluronidase on their membrane, and that breaks down the hyaluronic acid and allows the cells to no longer be quite so sticky. Now, to get through the zona pellucida requires a specific process known as the acrosome reaction. The acrosome is basically a single vesicle between the DNA of the sperm and the plasma membrane of the sperm. And it contains a lot of different enzymes, basically. And the acrosome reaction, you can think of it as basically being completely analogous to exocytosis. So if you think of this as a very big secretory vesicle, remember exocytosis is always initiated by an increase in calcium. And in this case, the acrosome reaction is initiated by a protein on the zona pellucida known as ZP3. There's a receptor for this protein on the sperm plasma membrane. So the interaction of this receptor with ZP3 initiates a signaling pathway that increases an acyl triphosphate inside the sperm. You get an increase in calcium, and the calcium causes fusion of the membrane of the acrosome and the plasma membrane. Now with exocytosis, when that happens, you get fusion of a small vesicle at a single point. So nothing else goes on except the contents are released. But in this case, you get fusion all of these membranes all around the vesicle. So you get fusion all over. And basically what happens is the plasma membrane and the outer acrosomal membrane disintegrate. That releases the contents of the acrosome and leaves the inner acrosomal membrane and the DNA of the sperm remaining. Now this process has three important effects. One, it releases enzymes, and this starts to break down the zona pellucida, but it's not the enzymes, and these enzymes themselves, they're not high enough concentration for a long enough period of time to get all the way through the zona pellucida. So it's basically making a little nick on the surface of the zona pellucida near the head of the sperm. It also activates these proteins on this end of the plasma membrane that still remains, and these are fusion proteins. And they're important for getting through the third barrier because they allow the sperm to fuse with the plasma membrane. And finally, it exposes this inner acrosomal membrane. Now there's enzymes on that membrane which will also break down the zona pellucida. So at this end of the process, you have a little nick in the zona pellucida, and you have the sperm head right here with those active enzymes, and you have the tail pushing it. So basically, as the tail pushes it through the zona pellucida, these enzymes break a little pathway through it and allow the sperm to pass through. Okay, any questions on that? Okay, so the final one is the getting through the ovum plasma membrane. And the first thing that happens is this inner acrosome membrane is kind of sticky. So once it gets through the zona pellucida, it sticks to this plasma membrane and brings this equatorial region down in contact with the plasma membrane of the ovum. And these fusion proteins then bind to the plasma membrane and cause fusion of the inner acrosomal membrane, actually the plasma membrane of the sperm and the plasma membrane of the ovum. And that initiates the process of taking the sperm DNA into the ovum. So there's a continued fusion, but you also get some phagocytic activity so that the head of the sperm becomes completely surrounded by an ovum plasma membrane and continues to break down. That releases the male pronucleus into the ovum. Eventually the tail and the mitochondria are taken in, but they're destroyed by processes inside the ovum. Now I'll just point out one other aspect of this. The ovum has a series of granules known as cortical granules, and they have another important function that we'll talk about in just a minute. Okay, so we've gotten fertilizations completed. This lists sort of a timeline of important events that occur immediately after fusion of the sperm membrane with the plasma membrane of the ovum. So very rapidly you get an increase in calcium inside the ovum, and this causes a process known as ulelamining. Ulelamining is just a fancy word for the plasma membrane of the ovum. So it causes what's known as hardening of that plasma membrane. Then a few minutes later, you get release of the cortical granules. They contain substances which cause hardening of the zona pellucida. Now, these two hardening processes, hardening is just a word that means it's hard for things to get in anymore. And in particular, these two processes prevent polyspermia. Because we've sort of focused on one sperm, the first one that makes it, but remember there's a whole bunch of other sperm trying to get through two, and if more than one fuses with the ovum, you're going to get polyspermia, and you're going to get 3X chromosomes instead of 2X, the normal ones, and that's lethal. So you definitely want to prevent that. And we know that this hardening of Lululemon occurs first, because if you look at the process of fertilization in vitro, not infrequently you'll see some of the sperm trapped between the zona pellucida and the plasma membrane of the ovum. So they've managed to get through in this period between the hardening of the plasma membrane and 2 or 3 minutes later when you get hardening of the zona pellucida. But after that 5-minute period, sperm can't even get through the zona pellucida. Okay, shortly thereafter, you get the second meiotic division in the ovum. Remember the ovum, the first meiotic division occurred at ovulation, but the second one doesn't occur until fertilization. So the ovum is actually sitting there with 2X chromosomes, and the second meiotic division gets it down to 1X, so the embryo has 2X, one from the male, one from the female. The sperm nucleus decondenses during this period, and so you get egg and sperm pronuclea, and within about 14 hours you get DNA synthesis and the first cleavage, and now you have an embryo instead of an ovum. Okay, the final step in what we're considering initiation of pregnancy is, oh, forgot about this. Okay, take a minute break. I'll give you about 30 minutes to think about that. Okay, everybody have a choice? I'll tell you my choice, and my choice is number 3. And I'll start with what's wrong with the other, what I think is potentially wrong with the other ones. This one should work because it will prevent the sperms from getting up. But remember, there's nothing particular unique about the smooth muscle in the oviduct or the uterus, so it's probably going to inhibit smooth muscle contractions throughout the body, which is going to mess up an awful lot of other systems. So that's probably going to have a lot of side effects. This drug, it won't work at all because it's active during the uterus, and you want to inhibit hyaluronidase activity up in the oviduct. And finally, this could theoretically work, but you never want to actively immunize anybody with a protein that's natural to their own body, because if you get antibodies to a ZP3 protein, it could well attack the ovum, all the ova that are in the ovary, and produce permanent infertility. This agonist, in theory, anyway, would bind to ZP3 receptors of the sperm. When they're in the uterus, you'll get that premature activation of the acrosome reaction, so any sperm that got up to the ovum wouldn't be able to get through the zona pellucida, and you prevent fertilization. Okay, final step in this process we're going to talk about is implantation. And first of all, there's a couple of temporal considerations to keep in mind. Fertilization has to occur within 24 hours of ovulation. If the ovum isn't fertilized within this period, it starts to degenerate and it can no longer be fertilized. Implantation occurs about 6 to 7 days after fertilization. And this is an important process because there are important events going on in both the developing embryo and uterus. So during that period, the embryo moves down the oviduct. It sits for a little bit at the junction between the uterus and the oviduct, and then it moves into the uterine lumen and floats in the lumen for a few days before implantation occurs. And during that process, the embryo is undergoing some rapid differentiation. It basically forms two different types of cells, the inner cell mass, that's going to become the developing fetus, and the trophoblast, which is the outer layer, sort of this salmon colored here. And the trophoblasts are the cells that are going to interact with the uterine endometrium during the process of implantation and help form the placenta. The other important thing that happens once it gets down into the lumen of the uterus is it hatches from the zona pellucida. Remember, that's that hard glycoprotein shell. Then it's protected, that embryo, as it moved down the oviduct and got into the uterus. But once it's there, you got to get out of it so the trophoblastic cells can interact with the endometrium. Now, at the same time, in the uterus, the main thing that's going on is you're going to get a structural differentiation of the endometrium that's going to allow for implantation. The uterus is also secreting glycogen and other nutrients to help maintain the embryo during this period. And one thing we discovered when we started doing extensive in vitro fertilization was for implantation to occur, the development of the embryo and the structural changes in the endometrium have to be in synchrony. So if you do an in vitro fertilization and you wait, let's say, 2 weeks, and you put that embryo into the uterus, it can't implant because the uterine endometrium is no longer at the right stage to allow implantation. So these two structures have to be in synchrony in terms of the development for implantation to occur. Now, basically what happens with implantation is the embryo attaches to the endometrium. The trophoblast cells actually invade the endometrium, releasing some enzymes and stuff, and interact with the cells in the placenta, the endometrium, to form the placenta. Now, the placenta is basically a combination of fetal and maternal membranes. One major structure that we'll talk about a lot in our next lecture is the chorion. And the placenta has two big functions. One, it allows a close apposition of the maternal and fetal circulations without any actual mixing. So that allows the exchange of materials between the maternal and fetal circulations. It allows nutrients and oxygen to get into the fetus, and it carries away waste material that are produced by the fetus during its development back into the mother. And also produces the important hormones of pregnancy that we'll talk about in our next lecture. Okay, any questions? Well, we spent all this time talking about fertilization. I'd like to take a few minutes, talk about the alternative, which is contraception. Now, this is just sort of a brief overview. You'll obviously learn a lot more about contraceptive methods in the pharmacology course. But they fall into actually 4 different steps or types of approaches. The first 3 are sort of traditional methods. There are barrier methods that prevent the sperm from getting up to the ovum by barriers. There are steps that are mainly hormonal that present fertilization, and then you can prevent implantation. Now, the barrier methods are probably the most familiar to us and have been used the longest. And those are temporarily barriers, such as condoms and diaphragms, and then more permanent, but not necessarily completely permanent, because they can be reversed. But those are surgical ones, tubal ligation and vasectomy. Now, in terms of preventing fertilization, as I said, the primary mechanisms are hormonal, and these are ways of delivering synthetic estrogens and progesterones. And there's a variety of ways. One that's been around since the 60s is the pill. We also now have vaginal rings, patches, and subcutaneous implants and injections, all of which are different methods for delivering contraceptive drugs. They have 2 important effects. One, and the most important of these is the progesterone primarily. The estrogens help progesterone's action because estrogen induces progesterone receptors. But remember, progesterone blocks the LH surge. So this is a classic mechanism by which the hormonal pill prevents ovulation. If you don't ovulate, you can't become pregnant. But they also inhibit sperm transport. Again, remember I said that progesterone is going to decrease smooth muscle contractions and increase the viscosity of cervical mucus, and that's going to prevent sperm from getting up to the ovum. Some IUDs also inhibit sperm transport and actually contain spermicides. For preventing implantation, there's two mechanisms. There's a classic IUD, or intrauterine device, and this is thought to produce an inflammatory response in the endometrial lining that prevents implantation. The other is the morning after pill. And the morning after pill is basically a high dose of the estrogen and progesterone that's in the normal pill. And it's thought to work by a couple of mechanisms. First, the high estrogen, if you take it after ovulation, early in the luteal phase, remember estrogen causes luteolysis, so it'll kill the corpus luteum before normal luteolysis, and that, since implantation occurs at the end of the luteal phase, that'll prevent implantation. It also contains a high amount of progesterone, which will have two effects. If you take it before the LH surge, it's going to block the LH surge. If you take it after, it'll advance the structural changes in the endometrial lining, so when the embryo does get down there, it's no longer in sync and implantation doesn't occur. So you remember I said there are 4 different methods. The fourth one is relatively new. It's only about 3 years old. But it makes use of a very old technique, which was called the rhythm method. The rhythm method, it used to be that women who didn't want to take medications would follow their basal body temperature, and the idea was progesterone increases your basal body temperature. So if you start taking your basal body temperature at menstruation, as long as it hasn't gone up, that's your period where you could get pregnant. But as soon as it goes up, that's basically 2 or 3 days after ovulation, and remember the embryo has to be fertilized within 24 hours. So then you could have intercourse between when it went up and your next menses. Now the problem with the old method was that minor fluctuations in basal body temperature can occur regardless of what progesterone is doing. For example, if a woman has a subclinical infection, she might have a minor increase in her basal body temperature. And if she had intercourse, then she'd become pregnant. So it was never recommended as a means of contraceptive. But this new one actually has been approved by the FDA, and it doesn't just use just a short period of temperature. It follows your basal body temperature over 3 or 4 periods and keeps track of it, and keeps track of when your menses occur, and then it can predict pretty accurately when you're going to ovulate. So it gives you periods during your upcoming menstrual cycle when you have a very low chance of getting pregnant, and it's safe to have sex, and periods when it's high. And it's about 96% effective. Okay, any questions on that? You want to keep going or take a brief break? How many people want to keep going? How many people want to take a brief break? Okay. 10-minute break, or you want to start at 1:45?